Hello, my name is Catherine Morrison, and today I will be presenting on signal transduction by G protein-coupled receptors, also known as GPCRs. This presentation fulfills the requirements for Chem 530, Concepts of Biochemistry, instructed by Dr. Sean Stacy, and is due July 1st, 2026. So let's get started. First, let's define signal transduction. Signal transduction is the process cells use to send messages across a cell membrane and trigger responses inside the cell, and the cell membrane has hydrophilic regions on both the extracellular and intracellular sides with a hydrophobic lipid layer in between. This lipid layer gives a membrane structure and stability while still allowing signals to be received and passed into the cell. In GPCR signaling, a ligand acts as a stimulus by binding to and activating the receptor. This activation begins a series of intracellular events or signaling cascade that can amplify the message and initiate important cellular processes. On this slide, we will continue explaining signal transduction by discussing why it is important. Cells need to receive signals in order to begin processes that affect the entire human body. If the body could not send messages across a cell membrane, essential cellular processes would not occur, and the body would not be able to function properly. Now let's look at some of the basic mechanics of signal transduction. The cell membrane contains many types of receptors, including GPCRs, which make up one of the largest receptor families in the human body. To activate these receptors, a stimulus is needed. Common stimuli include ligands such as hormones, enzymes, or other proteins. Mechanical forces can also act as stimuli, including fluid shear stress, membrane strain and tension, and hydrostatic pressure. Photoreceptors are another example of receptors that respond to stimulation, especially in or near the eye. Cellular damage can also activate receptors. When tissue damage occurs, proteins released during the healing response can stimulate receptors and signal the cell to begin repair processes. Transduction refers to the relay of molecular signals from one part of the cell to another. This process is similar to a relay race, where one runner passes a baton to the next until the signal reaches its final destination. When receptors are activated, they create a domino effect inside the cell. For example, GPCR activation causes a conformational change that allows the signal to continue into the cell. In ion channels, ligand binding can open a previously closed channel, allowing ions such as sodium, potassium, chloride, and calcium to move through. In receptor tyrosine kinases, stimulation causes two receptors to bind together in a process called dimerization, followed by ATP-driven cross-phosphorylation. These changes initiate signaling pathways that become amplified as the signal moves through the cell. Each pathway is designed to produce a specific response, such as activating processes in the endoplasmic reticulum or nucleus. Once the response is complete, the cell itself uses self-regulating mechanisms, such as negative feedback, to stop the reaction and return receptors to their inactive state. Now we're going to discuss the main types of membrane receptors. The first type is the receptor tyrosine kinase. This receptor is made of two separate parts. One portion contains the site where a ligand or protein binds, and once binding occurs, the two receptor parts come together in a process called dimerization. Beneath the cell membrane, this activates cross-phosphorylation using ATP, which allows the signal to continue inside the cell. The next type is the ion channel receptor. Ion channels are usually closed until they are stimulated by a ligand, protein, or physical force. When stimulated, the channel opens and allows ions to move across the membrane, either from the extracellular space into the cell or from the inside of the cell to the outside. Common ions involved include sodium, potassium, chloride, and calcium. Once equilibrium is reached, the stimulus is released and the channel closes again. Finally, we will discuss G protein-coupled receptors, or GPCRs. GPCRs are activated by ligands such as hormones, proteins, and enzymes. This receptor family will be the main focus of the following slides. In this slide, we are going to talk about a G protein-coupled receptor basic anatomy. The first thing that we're going to talk about is the actual cell membrane, which is a lipid bilayer that contains hydrophilic heads and hydrophobic tails that are lying back to back. The hydrophilic molecules, or the heads, allow for fluidic movement throughout the outer and intracellular membrane. The middle portion actually gives stability so that it keeps together in a membrane rather than it breaking apart, going everywhere. Number 2 is the actual 7-membrane alpha helical GPCR. They call it this because this particular membrane is interwoven within the cellular membrane. The head, which is on the outside of the cell membrane, is where it will be stimulated, and then the inside, it has a tail that is actually attached to the G proteins. Number 3 is an actual ligand, which could be any kind of stimulus, such as a hormone, an enzyme, or other mechanical or photoreceptor stimulus. Number 4 is the receptor that is the same shape as a ligand, so that when they come together, it forms a lock and key model where they fit perfectly and will activate the molecule. Number 5 is the alpha subunit, and on this alpha subunit is attached a small GDP molecule. Number 6 is a gamma subunit. Number 7 is a beta subunit. All these subunits are known as a heterotrimeric G protein. Number 9 occurs after the conversion of GDP to GTP when it becomes activated. The cell becomes activated when the ligand fits into the receptor. Once this is activated, it forms a conformational change within the G protein. The alpha beta subunit moves to one side, and the GTP alpha subunit moves to another. On this slide, we will be talking about the activated G alpha sites. We are going to enlarge the GTP alpha site so we can talk about it. But before I begin that portion, I want to touch on the gamma beta subunits. These are still a very important product, and what they do is they stay along the cell membrane, and they have 3 functions. They regulate the ion channels. They modulate the enzymes such as PLCs and the adenyl cyclase, which are used further on in the G subunits activation pathways. And it also are mediators of the G protein-coupled receptor kinases. And then once it does that, it will go back and wait for the inactivated alpha GDP. So let's move on. The first one is a G alpha S, which is actually a G alpha S, which means stimulator. The next one is a G alpha I, and the I stands for inhibitor. And this actually will regulate the predominance of whether the cycle will continue to make ATP to cyclic AMP. Then we have the G alpha Q, which is an enzyme activation pathway. And then we have one more. This particular subunit is not often referred to because it is not as well known as the other pathways, and that is the G alpha subunit 12 and G alpha subunit 13. Pathway that we are going to touch on is not necessarily the first one that activates, but it is a G alpha A I subunit. And this one, as you can tell by the tail, has a stop modulator. The next one is a G alpha S, which is a G alpha S stimulator. This will go on and go and stimulate the adenyl cyclase. This reaction will then cause the ATP, which is in the intracellular membrane floating around, and it will take it and it will convert it to cyclic AMP. The cyclic AMP then goes through a process of cleaving and activating with pKa, which will then phosphorylate target proteins. And those are the cellular responses that occur. The next one is a G alpha enzyme activation. In this one, we have the PIP2, and it will be cleaved with the reaction of a PLC. Once it is cleaved into two subunits. The first subunit is the IP3. The IP3 then activates the PKC. The PKC then activates within the nuclear cell calcium, and this causes cellular responses. One of the cellular responses is, and the second protein is DAG. And then a secondary response from the calcium on the inside will actually release excess PKC, and the PKC will attach to the DAG, and this will cause another secondary membrane to produce more pathways and stimulus to proteins along the line. This slide may look busy, but I chose it because it provides a good example of an activated G alpha subunit 12, 13. G alpha 12 and G alpha 13 are two related components that can activate several downstream pathways, as you can see from this picture. These pathways can influence cellular responses such as cytoskeletal organization, cell growth, and gene transcription. Compared with the G alpha S, G alpha I, and G alpha Q pathways, G alpha subunit 1213 signaling is discussed less often and is still an active area of research. However, advances in technology have increased scientific interest in this pathway because it may be involved in cancer-related or oncogenic responses. We will look at disease states associated with GPCR signaling. This slide provides a broad overview of several major disease categories. The first category is a metabolic disorder, which includes obesity, type 2 diabetes, cardiovascular disease, and osteoporosis. With this particular set, it will hit things such as the ghrelin, GHSR, the parathyroid hormone, and then the next order is the immunological disease, such as allergies and autoimmune disease. And one of the pathways it attacks is the actual histamine. For the next one is the neurodegenerative diseases, such as Alzheimer's disease, Parkinson's disease, Huntington's disease. And the pathway that it disrupts here will be like the serotonin, dopamine, and glutamate. The next one is infectious disease, which include HIV-1, Chagas disease, CMV, which is cytomegalovirus, or EBV, which is the Epstein-Barr virus. And with these particular ones, it's like the HIV-1 viral GP120, or the CXCL17, or the hedgehog protein. And the next one is the cancer, which includes ovarian cancer, lung cancer, head and neck, and small cell sarcomas. This slide provides an overview of GPCRs that can be used in treatment strategies. Because GPCRs are found throughout the membrane and are involved in many important cellular processes, they are major targets for drug therapies. Drugs can also be designed to bind specific GPCR sites that are involved in certain disorders, allowing treatments to be more targeted rather than broadly affecting the entire body. GPCR-based therapies may also focus on the signaling pathways that occur after receptor activation. In the future, drug development may continue to move beyond simply targeting the receptor at the cell membrane and may also focus on intracellular signaling pathways. This could help create more precise treatments with fewer unwanted side effects. In this slide, we will review examples of drug therapies and their GPCR targets. The chart shows the disease state, the drug that is related to the GPCR target, and the main site where that target is active. This is only an overview, so it does not include every possible disease state, receptor, or treatment site. For obesity, the GPCR target shown is the GHSR, which is associated with the hypothalamus and pituitary gland. This one will suppress hunger. For type 2 diabetes, the target is a GLP-1 receptor, which is mainly associated with the pancreas. And this allows for the pancreas to be able to uptake glycogen at a better rate. Then we have the cardiovascular disease, and the drug would be the adenosine receptor, and its site would be the coronary receptors, which are obviously on the heart. Then we have the osteoporosis, and it will be the PTH receptor, because the PTH is a major contributor to bone growth, and this will affect bone and kidney. Then we have the allergen, and it will be a histamine H4 receptor. This will be used whenever we have leukocytes. The leukocyte family has 3 types: the neutrophils, basophilics, and eosinophils. These 3 all have specific jobs to do. The one that would be involved with the allergy would be the eosinophilia, which would occur when an allergen response is occurring. The next one are the neurodegenerative diseases, such as Alzheimer's disease, which uses a drug which is CXCR2, and it will affect the hippocampus. Parkinson's disease is a muscarine acetylcholine receptor, and this will be on the basal ganglia. The next one is Huntington's disease, and its form will be the MGLUR4, and this will affect the stratum. AIDS uses the CCR5, and with this group, it's actually not a targeted site, but the actual cells that have gone out of control. So it would be on a cancerous cells. Melanoma uses CXCR4, and it also affects cancerous cells. Breast cancer, CXCR2, which affects the cancerous cells. Advanced and or metastatic solid tumors, and these are using on the smoothened area, and these are also on the cancerous cells. One thing that I found interesting was that for Alzheimer's disease, they use the same drug type as breast cancer, which is CXCR2. But because they are targeted at different areas, they do have a different job. In summary, the G protein-coupled receptors are one of the largest and most important receptor families found in the cell membrane. The GPCRs are known as the 7-transmembrane alpha helix protein that weaves through the membrane and allows the cells to respond to a wide range of stimuli, including hormones, enzymes, light-sensitive receptors, mechanical forces, and signals related to cellular damage. Once activated, the G-alpha subunits begin a signaling cascade that activates second messengers and other intracellular components, such as the pathways involving the endoreticulum inside the nucleus. It also causes any kind of damages to the GPCR or its pathways will cause disease states such as the ones that we covered as obesity or HIV-1 or ovarian cancer. But luckily, there is a large amount of drug therapies being developed because of the advances in technology and in computer manipulations in computational chemistry. Also, because it is a targeted drug therapy, they can now go specifically to the GPCR portion that is being damaged and just target that one area. This way, instead of giving it a general type of drug that can cause more harm than good because the side effects are so bad, that now they can just target a certain aspect of the pathway to give it a better chance of cure. The following 3 slides are all references that are included within this presentation. I would like to take this time to thank you and have a good evening.